In this video, we'll discuss sarcomeres. Now, a mere is like a unit of something. And sarco, we mentioned before, means flesh or meat. Basically, it means muscle. So a sarcomere is a unit of muscle. Just based on that name, sarcomeres must be pretty important. And yeah, I mean, they're very important. So let's look at them. Revisiting this wild diagram of excitation-contraction coupling, we'll remind ourselves that when our brain decided we wanted to voluntarily move some skeletal muscle, we initiated this series of cascading events that eventually flooded the muscle cell with calcium. But why? Well, the secret to understanding that lies in striations, but really, we'll see that striations, if you look more closely at them, are organized into little subunits, little sarcomeres. So understanding sarcomeres is the key. But let's slow down for one slide and look at what are striations. Well, here's a single myofibril. And remember, each muscle cell is packed with hundreds to thousands of these myofibrils. The arrangement of these myofibrils, like the stuff that they're made out of, is perfectly aligned. Meaning, if here is one muscle cell, the striations run the length of the muscle fiber, the muscle cell. So if we said hundreds of thousands, you'd have to draw hundreds or thousands of lines within this muscle cell. But everything inside those lines, those myofibrils, is perfectly aligned. Meaning that there's some dark stuff in them and some light stuff and dark stuff and light stuff. And all the dark stuff perfectly lines up and all the light areas perfectly line up. So you get these striations throughout the cell. These dark regions within the myofibril are called A-bands. And let's note that the word dark has an A in it. So this is an A-band. And here is another A-band. And here is a third A-band. There are also I-bands, the lighter colored bands, and let's note that the word light has an I in it. So the dark bands are A-bands, and the light bands are the I-bands. Here's an I-band, here's an I-band. So what does this have to do with sarcomeres? Well, sarcomeres are these units which just repeat over and over and over down the length of the myofibril. So if you find the I-band and you look right in the middle of it, you'll see this zigzagged structure called the Z-disc. And then if you find another Z-disc in the next I-band, what you've done is you've captured a muscle unit, sarcomere. This region will repeat in both directions again and again and again, just copies of this sarcomere, thousands or tens of thousands of times down the length of that muscle fiber. So if we can understand what happens in a single sarcomere and multiply that thousands and thousands of times, that's muscle contraction. As a very earliest approach to this concept, look at my fingers. Is there a region that you would call the A-band? Hopefully, perhaps, here in the middle where my fingertips are found, that is thicker. There's more fingers there. That is arguably a darker region. That would be like an A-band. And on each side of that would be I-bands. And in muscle contraction, what will happen is that things will slide over each other. Remember the sliding filament theory? And the sarcomere will shorten. I mean, on a subcellular scale, a very small amount of shortening. But you do that 80,000 times down the length of a muscle fiber, and you do that in a bunch of fibers within the cell, you can get all kinds of muscle contractions. And not just my waving arms, but also the expressions on my face. All done by sarcomeres contracting. So what are in sarcomeres? Myofilaments, there are thick ones and thin ones. So the thick myofilaments, kind of a fancy way of saying these muscular filaments that are made of protein, are myosin. And then the thin myofilaments, these thinner muscular filaments that are made of protein, are called actin. So myosin and actin are found within the sarcomere in a very particular arrangement. If we look again at the A band, we'll see that there are both red and blue lines on this diagram. And the thicker ones that happen to be red are myosin. The thinner ones are actin. And then if you look in the I band, you'll see only actin. We'll look at that again as we start to name some of the other structures on the sarcomere. But here's a quick look at myosin, and here's actin. It's tough to tell here, but myosin is a much thicker, bulkier protein. Okay, so looking more at this sarcomere, it's a slightly different diagram, so let's get our bearings. Sarcomeres always start and end at those Z lines, or also known as Z discs. Those are in the middle of the I bands. Here's one sarcomere. Anywhere you see this thick myofilament in blue in this image, that's myosin. But I can also see here mixed in with that myosin is actin. So the A band has both myosin and very small thin actin filaments mixed together. But in some regions, there's only myosin. So in the A band, what do we call the region that has only myosin? That's the H band. What do we call the very middle, where these myosins are all anchored together? That's the M line. Z discs we already discussed. A bands and I bands we've discussed. Yeah, we're looking good. So that's just some basic geography of a sarcomere. Mostly what we're interested in though is the regions of the sarcomere in which the myosin heads, see the little bumps on the myosin here, come into contact with the actin, because that's where the muscle contraction really happens. So let's look more closely. Here's a view of myosin. We can see that a single myosin molecule has a long tail, a little hinge region, and then a globular head. And if you can't quite tell, there's actually two globular heads on the end of this myosin molecule. Bunches of those myosin molecules are all wrapped together. If you look at an actin molecule made of these actin subunits, it's like two strings of pearls wound around each other. That's our actin. And the myosin heads really want to grab onto the actin at the active site. And when your muscles are contracting, what's basically happening is that myosin head is accessing the active site and it's pulling on it. But there are some other proteins, accessory proteins called troponin and tropomyosin, that are going to have something to say about whether or not that myosin head can attach to actin. So let's look at that on the next slide. Here we're going to find out whether or not the myosin heads can pull on the actin. And they do that in both directions, by the way. So those myosin heads, they want to attach to the active site on the actin molecule, but they can only do it when calcium is present. So finally, we're understanding why, when I think about contracting a voluntary skeletal muscle, why does that cause calcium to flood into my muscle cells? The answer is because calcium will bind with one of the accessory proteins called troponin. and it will cause a structural shift that will then cause a change in the other accessory protein here, tropomyosin, which will shift aside. Because right now that tropomyosin, this kind of green cable, is blocking the active site. And you know who's keeping it there? Troponin. Troponin is like the glue or the clamp. The clamp, that's better. It's a clamp that's holding tropomyosin in place. If you unscrew the clamp with calcium, then tropomyosin will shift out of the way, and then the myosin head can grab the active site and start to contract the muscle. And it'll keep doing that until either there's no more calcium or there's no more ATP for energy to cause the myosin head to pull, 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 pull. You probably won't run out of ATP anytime soon, but you might run out of calcium if you're thinking contract, 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 I'm flexing, I'm flexing, I'm flexing. My brain is firing a signal that's causing the calcium to flood the cell. But as soon as I decide, no, stop contracting my muscles, then that whole cascade of events stops. The calcium goes back into the sarcoplasmic reticulum, and there's none there. And then tropomyosin will get clamped back into place. No muscle contraction will occur. Which is basically what's discussed here. So that's what happens in just one single sarcomere. You need calcium, you need ATP, and then the myosin heads will pull on actin, causing contraction, in this case, flexion of the elbow. And if you look closely at a sarcomere, you'll see these Z discs will slide closer to each other during that contraction. And that explains, at an appropriate level of detail, how muscle contractions work according to the sliding filament theory of muscle contraction. We so smart now.